Yeah, so we started about archiving of electronic raw data in the GxP environment. So also for BASF an important and interesting topic. And yeah, looking forward to your talk, Mr. Burkhard. Thanks. Yeah.
Thank you. I'd like to share my screen here.
You should be able to share it.
Yep. Let me just share my screen. Okay. Thank you for the kind introduction. I'd like to present in my talk today the use of the ADF format for archiving purposes in the GxP production environment. I'm part of the analytical group located in early drug substance development in Biberach, Boehringer Ingelheim, Biberach. And when you take a look at the timeline for chemical production, for API production, we are located here. In the early phase, we are taking over the projects from Medchem, from research, and bringing those projects through late lead optimization phase, preclinical phase, phase 1, up to phase 2, where late drug substance development will take over. We are supporting the productions of larger amounts of active substance in the pilot plants. We are developing scalable and robust processes for early development work packages. We provide in the IPT lab, in the process testing lab, analytical support for release of all self-manufactured intermediates up to phase 2. We are supporting the process research units with tox material, and we implement the first kilogram campaigns in the kilo lab with GxP, GMP batch release. So when we start analytics here, we have a non-GxP process through clinical phase and phase 1, and at least at the end of phase 1, we are bound to the GxP process for release of these phase 1 API starting materials and API materials. Of course, overview about the IPT lab. This is an actual view of our laboratory, two of my co-workers here. And as you can see, it's a fairly large facility because we need a lot of analytical instruments and a lot of analytical techniques throughout the development of these final APIs. We have some analytical methods or techniques that we can provide to our customers. And as you can see here from the number of systems, most of those are liquid chromatography based. Liquid chromatography, MS detection, supercritical fluid chromatography, gas chromatography. We have 7 different instrument vendors. Each one is specializing in one of these analytic techniques. And these 7 vendors that we have instruments of in our lab manage to provide 8 different electronic file formats that we have to handle throughout all the data handling process. For when it comes to handling electronic raw data, the day-to-day handling is usually done by the chromatographic data systems or vendor-specific formats, vendor-specific software. But when it comes to backup and archiving, there are different regulations that we have to follow. First, the Boehringer Ingelheim regulation, they state that the archiving format should preferably be standardized, non-proprietary or an open format. That's something we try to achieve with an Allotrope file format. And the archiving period that we have to take into consideration is 30 years for GMP and GLP data. The MHRA is a European authority that states that raw data are original records or a true copy. Original records is defined as the data that have been at the first time recorded by an analytical instrument to a non-volatile memory, so something like an SSD or a hard drive. A true copy is a complete copy of the original records that can be handled. We can produce as many true copies as we want. We can transfer them, we can delete them. We only have to take into account that we do not delete the last true copy, of course. Data archive arrangements must be designed to permit recoverability and readability. and in case of legacy systems, the ability to review data periodically. This ability to review is one of our major concerns here for legacy systems, because it does not only contain a data archive, so we don't need to just take our data in a vendor-specific file format and put it into an archive for 30 years and let them stay there. We have to review this periodically, and review means we have to dig into that electronic raw data. We have to use the original vendor application to open that raw data and check periodically if this is still possible to review this data over a 30-year of times. Backup and recovery process should be validated and periodically tested, and each backup should be verified to ensure it has a function correctly. That's basic GMP functionality, of course. The FDA, however, has a very unique view of the term backup. They state that a temporary backup copy, e.g. in case of a computer crash or other interruption, would not satisfy the requirement to 11.68 to maintain a backup file of data. That means that a backup, what we are doing in our lab from day to day, is not considered a backup according to the FDA regulations, because these backups are mentioned for computer crash and recovery of a system in case of a computer crash. The FDA defines a backup only a backup of a long-term storage. So in case of an archive, that's a backup of an archive. The FDA does not require us to use day-to-day backup copies. Interesting that the European authorities and the FDA share a pharmaceutical mutual recognition agreement, which means they accept vice versa each regulations they set up and each audit result from auditors. Some considerations from these regulations. Do we need a temporary backup at all? Is a backup in an archive identical by means? Because if we can omit a backup, that would make data handling for us much more convenient. Do we need or want a living data archive or a black hole? A black hole has only one entry and presumably no exit. A living data archive helps to keep the data active, helps to keep the data accessible throughout the whole 30 years lifetime. The number of different file formats for a single batch release that we perform in our group for one batch release report is 3 to 5, which means we have 3 to 5 different analytical techniques for a batch release, for example, solubility testing or water content or HPLC or something like that. And we have to handle all of these 3 to 5 different file formats in one batch report. Raw data handling is complicated because data is locked either in a vendor-specific application or even in an encrypted database. In the case of an audit, we have to take into consideration that we need to access all of these 3 to 5 different analytical techniques just to show the auditor, for example, a simple thing like an audit trail. How do we perform data handling during decommissioning of retired legacy systems? These legacy systems accumulate data for over a decade in our lab and now have to be retired. The only thing usually we can do here is to shift the whole database to an archive, but that does not contain the readability or the review that that is required by the authorities. So in some case, we need to address these limited access to our files in risk assessments or in shifting these data to another archiving format. What do we do with the recorded data in an archive except of preservation? That points to the usability of generated data in future systems. Maybe more on that later in the presentation. To summarize, it's time we shift from to a data-centric mindset that recognizes that apps may come and go, but our data source is our primary asset. So we don't want an application-centric approach as we have it today. We want a more data-centric mindset for using of data in future. Before we decided to use the Allotrope file format, we took a look at other file formats that are on the market or that we already have in our facility. The UPAC JCAM DX file, of course, a very old file format used since 1993. We have lots of systems that can export data in JChem-DX. Unfortunately, this is not the very best format when it comes to GxP because there's no metadata information, there's no audit trail, and we do not have a closed container with a checksum that can prove that all the data inside of that container is untampered, unaltered. SILA, the SILA 2, and Pistoia Alliance Unified Data Model. These are some pretty promising file formats. At that time, we looked at them some years ago. The user base was unfortunately very, very limited, and we did not find a vendor that provided an export functionality in their software in any of these file formats. OPC UA, of course, that's an architecture or file format that we have in use in our industrial automation systems, process control systems in the pilot plant, Siemens SPS controls, for example. And the latest initiatives using these data for laboratory systems, the last initiative is a very promising approach. We are watching that closely. Animal, that's something we heard about today also. From my point of view, it's some kind of an historic file format that had a considerable user base some years ago, but today this user base is limited, and also it's an XML-based file format which shares the same problems like the JChem-DX. So it's an XML file which does not contain a closed container and a checksum, and so for GxP use, we have to take into consideration of validating each of these individual steps, which is a pretty large task. For our archiving system, we have, for laboratory systems, we have the so-called DALIA application. That's a Boehringer name for Data Archiving and Holistic Lab Information Application. A course overview about that. Usually when you have a lab instrument controlled by a lab PC, you install a client, or we install a client on that lab PC that watches one local directory, and each file that appears in that local directory will be taken by that watcher software into the archiving system. We have an event-based file monitoring on that directory. And the DALIA system can provide an audit trail, it can provide metadata, it has a versioning system, it has a post-processing workflow for data conversion, data packaging, and finally ingestion to an archive. This is a validated system and a validated process for GxP data on laboratory systems. However, when we started to use the ADF file directly from the instrument, we can omit all of these workflow that we have in the DALIA system because the ADF files already contain an audit trail. They contain the metadata. They have kind of a versioning system. We don't need a data conversion because it's already done, and data packaging also. The final step for ingestion to archive is still present. But this greatly simplifies the workflow that we have for standard laboratory system in a GxP environment. For the use case, some years ago, when we decided to look deeper into the Allotrope file format, I went to all system vendors that we have instruments of in our laboratory and asked them about an export functionality to the Allotrope file format, and the only one who responded was Agilent at that time. Agilent was developing an Allotrope export plugin for their software, and we set up a small cooperation. So we provided demo data and test cases to Agilent, and they provided software versions to us to test. And that finally resulted in an export plugin version 1.3, which has been released to the market in 2023. So that's a product that you can actually buy today. It integrates into the Agilent software here. This is from the Open Lab chromatographic data system, the data processing tab. You have your sequence here. That's a demo sequence. You get this export ADF button in the software. After processing, you hit the button and you can export your ADF file to any file system you want on that system. It's a manual process today. So the automated function will come in future versions, so that after the processing here, the reprocessing, the PDF report and the ADF file are generated automatically and transferred to that shared directory I mentioned earlier in the DALIA system and directly ingested into our archiving system. Here's an overview about the systems that this Allotrope export plugin from Agilent will work on. It's the software here, it's the OpenLab CDS workstation and the client server system. Also the ChemStation version C, which provides, which uses the ACML file format, the Agilent Common Analytical Markup Language. The older versions of ChemStation A and B, which have been around since 1 or 2 decades, do not use the ACML file format, but you can still read your historic data into this version C of the ChemStation or even the OpenLab system, convert it to ACML and export it as ADF also. Exemplary hardware, that's basically the hardware that this plugin has been tested on in our laboratory. That's hardware we have actually in our laboratory, the Infinity 1260 and 1290 systems, LC systems, the DRD detector. We have a single quad mass spectrometer detector and an electronic light scattering detector. We have the SFC system over here, the supercritical chromatography system, and the GCs from the 7900 series with this rotary injector over here. I'm pretty sure that Agilent in the meantime extended the support for other hardware platforms and other modules. So you can most probably use that export plugin on different kind of systems today. What other vendors are supported? Surprisingly for me, Waters Empower is fully supported. There's a white paper on the Agilent webpage describing that process. So if you have Waters Empower software data and want to convert it to Allotrope, you can use the Allotrope converter plugin from Agilent. I'm not sure if Waters already has an Allotrope export plugin. Maybe they have it. We don't use that software in our lab, so I'm not familiar with that. Other vendors is a basic support. Basic support is something like you can take the vendor-specific raw data, vendor-specific binary raw data, and convert it into an Allotrope file. You get support of the data package containing all these vendor-specific files. But if there are no models, obviously you don't get any information about models in the data cubes. So the batch conversion option might be a very valuable option for the future. Batch conversion is a script that can be integrated, for example, in our Dahlia system as a post-processing workflow. And this simply takes older ADF files like the basic files I mentioned over here. When there are new models in the future and new versions released by the Allotrope Foundation or by Agilent from this export plugin, to have a background process and automatically convert older ADF files to the new ADF standard. The main reason we have been using that Allotrope file format is the viewer software. So when we generate Allotrope files, we also want to review it. And there are 2 major options for reviewing Allotrope files, which is one from the HDF consortium and the Allotrope Foundation, of course, the Allotrope viewer. But to be honest, both of those are pretty basic. They offer pretty basic functionality. That's why Agilent generated this Chrome viewer for the Allotrope files. This is an internal project, has been used by Agilent internally just to review the files they generated with the export plugin. And this is not a software you can buy, but it has provided by Agilent to us for reviewing our generated Allotrope files. We have some information here about versioning, some information about the sequence runs. In this case, it's only a demo sequence with 3 runs, 2 blank injections, 1 system suitability test. Here's some information about metadata, the name of the analyst, injection date, injection volume. Here's some information about the detector, chromatogram, DAD view, the UV spectra. Here we find some information about the system itself, consisting of these 4 modules, serial numbers, firmware versions, and so on. Here a view of the data package inside the Allotrope file. Here we have the peak table that we usually use as a report, retention times of all of our peaks, relative peak area, and some other information. Here's the chromatogram that is shown here from the peak table. You can see some peaks. This is a basic, very basic viewer, so we can zoom in and out maybe to see these smaller peaks here, and we can highlight one peak, and this highlighted peak shows the UV spectrum over here. Most interesting for us are these export audit trail functionalities here. We can click here on this button and see the audit trail from the Allotrope file that is generated upon generation of the Allotrope file. We can also see the run audit trail and the activity log, which is the main audit trail directly from the measurement here. So this will be extracted from the data package and shown in human readable format here. When we have auditors in our facility that want to see electronic raw data, usually the first question is, show me the audit trail. And this greatly simplifies the process. So we don't need to go into the original file format. If the auditor wants to go into the original file format, we have this extract button over here. This extracts the contents of the vendor-specific binary files to our file system, and this can be reopened with the vendor-specific application, that's ChemStation and OpenLab in our case. Looking into the future, some considerations that we already heard today from other presenters. What we need for the future is more instrument vendor support. This is the basic functionality we need to have. We need more models. We need more instrument vendors that directly support an export of Allotrope file formats. We also need a universal viewer tool for visualization that provides a more holistic data view. The Agilent viewer that I showed in the last slides is a very convenient tool. And it's a very nice tool for LC-MS data. But as I mentioned earlier, we need 3 to 5 different analytical techniques for one batch release report that we generate. And it would be very convenient to have a viewer tool for visualization of all of these data in 1 or 2 screens. We need functionalities for open data check, which is mandatory for GxP processing. Some other things we also heard today, ASM export for non-GxP cloud systems. That's something I think all of the larger pharmaceutical companies are working on, some kind of a cloud system that provides support that provides access to all analytical data or process data that is generated. ADF is too bulky for the daily use. I've shown you a demo sequence with 3 sequence lines, and that ends up in a file, in an ADF file from 30 to 40 megabytes. When we have a sequence line for a production run, for a GMP sequence that has 20 to 25 sequence lines, and that results in an ADF file from 600 to 800 megabytes. We have 18 systems in our lab. We have more than 300 in our area and thousands in our company. This ADF file that we produce here is far too large for everyday's use and for storage on a cloud LIMS system. We also need ASM extractor and converter from ADF for non-GxP use, so that we can access our ADF archive that we have. Our Dahlia archiving system is generating large amounts of ADF or accumulating large amounts of ADF files. And we want to access those or access some contents of those. And here the ASM file is a perfect alternative for the non-GxP use. Also, batch ADF and ASM converter tool for decommissioning of legacy systems. That's something that would be very convenient because we have an increasing number of legacy systems that have GxP data on them, and we need to extract these data in a vendor-independent file format. So ADF and ASM batch converter, which is not a manual process but an automated process, would be a highly regarded tool. And far in the future, advanced analytics and AI techniques might help to extract insights from diverse data formats that uncover patterns, trends, and correlations that might not be apparent through a traditional analysis method. That's something that is proposed by some other speakers today also. That's something I can agree on. With that, I'd like to conclude my presentation. Thank you very much to Heiko Fessenmeier and his team from Agilent for providing us support for the ADF export plugin and the viewer tool. And thanks for all of you for attending the presentation. Thank you.